Yes, yes. Yeah. ಕೇಳ್ತದ?
Good afternoon, everyone. I, Dr. Sushrutake, welcome everyone on behalf of Yenepoya Homoeopathic Medical College and Hospital, Mangalore, for this national webinar. Now, I request our Dean, Dr. Vivekananda V. Varnekar, to welcome the gathering.
Good afternoon to all of you. On behalf of Yenepoya deemed to be university, on the behalf of the faculty of homeopathy, the Vice Chancellor, Chancellor, Pro-Chancellor and Registrar, I welcome all the delegates who have responded to our national webinar series in homeopathy, which is commencing from now, wherein I would like to give the background of the university. University established in 2008. It is deemed to be university with NAC accreditation, which is in first 25 universities accredited by University Grant Commission. Well, what we find over here is we have got a medical fraternity, we have got a dental faculty, they have got pharmacy, Ayurveda, homeopathy, naturopathy. Then we have got physiotherapy, nursing and other paramedical and a well-established, nationally reputed research center. We are just about 2 years old to begin with the BHMS course, and wherein we thought of taking an advantage of this COVID situation and commencing with the national webinar series. Today's our guest speaker is Dr. Jyotish. He is a professor in pharmacy who will be enlightening you about the basics of homeopathic pharmacy. We have got series of lecture in first phase. We are organizing 6 webinars, which will be quite useful to you. I think all the delegates, you have responded very well to us. I think we'll be in position to gather much about this. And Yenepoya University always will be focusing on the extra knowledge as enhanced knowledge with the innovation and research in the field of homeopathy in a real section, as we manage with the research program from the faculty side. Also, we manage the research program for the student base. Ultimately, our idea is to contribute something from the homeopathy from this institution as well as from this university. Our university is having a different campuses, and homeopathic college is one of the fully equipped with a 25-bed hospital with a lot of outreach sections and the research section, which will be definitely an experience for any of the homeopaths to graduate from here. And one more happy news which I'd like to highlight is we already started with the admissions for the PhD program also. wherein I would like to once again welcome all of you, wish you all the best, and you being with us and you give your valuable suggestion. Thank you. Thank you, Dr. Shushruth. I would like to continue with the seminar. Thank you.
Thank you, sir. Now, I request Dr. Apeksha, Assistant Professor, Department of Mathematics, to introduce the speaker.
Good afternoon, everyone. I believe it's my privilege to introduce the resource person of today's national webinar, Dr. A.B. Ramjyothi, sir. Sir attained his BHMS degree from Vinayaka Mission Homeopathic Medical College, Salem, in the year 2003, and his master degree in homeopathic pharmacy from the same college in the year 2006. His teaching experience. Sir has been teaching and he has a teaching experience of more than 13 years for both undergraduate as well as postgraduate students. His administrative experience. Sir is the head of Department of Homeopathic Pharmacy in Athurashram Homeopathic Medical College since May 2019. Sir has hands-on training in various fields such as advanced biostatistics, faculty training program, animal experimentation, signal detection and casualty assessment, statistical reasoning and algorithms in pharmacovigilance. Sir secured first rank in MD examination conducted by Vinayaka Mission University, Salem in the year 2006 and was awarded gold medal. Sir has been the primary investigator for research project on the study of antioxidant activity of homeopathic drugs used in arthritis, which was funded by Rajiv Gandhi University of Health Science, Bangalore. He is appointed as a co-investigator for CCRH, Drug Proving Unit at National Homeopathic Research Institute in Mental Health, Kottayam. He is a research guide for STSH program of CCRH. Sir is a peer reviewer in the International Journal of Homeopathy and Natural Sciences and Journal of Evidence-Based Complementary and Alternative Medicine. He is also appointed as a medical inspector by CCH, subject expert in interview board of Kerala Public Service Commission, and question paper setter in various universities of India. He also has the credentials of being the resource person in various faculty training programs and CMEs. It's indeed an honor to have you on board, sir. Thank you.
Thank you, Dr. Apeksha. Now, I request all the participants to fill the feedback form, which will be given at the end of the webinar. Okay, and to put the queries in the chat box during the webinar, and it will be answered at the end of the webinar. Now, I request our esteemed speaker to take over the session. Over to you, sir.
Thank you, Dr. Shushruta. And a very good evening to all. Respected Dean, coordinator and organizer of this webinar, and the fellow participants. So let me share my screen.
So any problems with the presentation?
To share.
Sir, open the PPT. Don't minimize the PPT.
Okay.
And then try to share the PPT. So you can close all the presentations once and again open the presentation. Don't minimize that. And then you can go to Teams app and from there you can present.
Okay. I have to close all the windows now.
Sir, I think you are presenting something already. It is showing us.
Is it visible now?
Not yet, sir, not yet.
My screen is visible now.
One second, sir. Nothing yet. Yeah, sir. Yeah, it's visible now.
Visible now. Okay. 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 Good evening once again. Today's topic is basics of homeopathic pharmacy. I was asked to take some relevant topic in homeopathic pharmacy. And why I took homeopathic pharmacy, the basics. We need to know the basics of homeopathic pharmacy, not only for a clinical practice, but also for the future of homeopathy, the homeopathic research. Homeopathic pharmacy, as you know, it is a science and art of identification, collection, preservation, standardization, preparations of homeopathic medicines, and also it includes the legal aspects of dealing with medicines. So that is why homeopathic pharmacy is a interdisciplinary subject which consists of biological sciences like botany, zoology, etc. Material sciences like physics, chemistry. Even it includes technology. Many of the aspects of technology is used in pharmacy, and also it utilizes the tenets of law, the legal aspects. So it is ultimately a interdisciplinary subject. Now, in pharmacy, it is mainly dealing with the preparation of medicines. As you know, it prepared the medicine from the drug substance to the drug. Then from the drug we prepare medicine, then ultimately the medicine is converted into remedy. So the process of identifying, process of selecting the drug substance, and that is what is called as pharmacognosy. It is about the identification of drug substance. Even it deals with the details of the medicine, the medicinal substances, the identification features, the habitat from where it has to be taken. So all these comprise in pharmacognosy. Now pharmaceutics is about the manufacturing of medicine. So from the drug substance, that is the raw material. From the raw material, the drug is prepared, and drug is, all of you know, it is a pharmaceutically prepared substance in crude form, and it is a substance which are capable of changing the sensations and function. Even it causes some structural changes also. So that is what we call as a drug. Now, when it comes to medicine, as you all know, it's a substance which is pharmaceutically prepared, but it should be potentized and it should prove in the healthy human beings. So that is what we call as medicine. So a transformation from a drug to medicines, what is called as dynamization or potentizations. So now another thing is the drug discovery. I will detail in next few slides what do you mean by drug discovery. Okay. Now, when this medicine is prescribed on the basis of law of similia, and when it is indicated, then it is known as a remedy. So this is a transformation of a drug substance to a remedy, and this is the core aspects of pharmacy. Now coming to the sources, I told you the drug substances are prepared from different sources. It can be from botanical sources, it can be from zoological sources, it can be from minerals or chemicals, or it can be from biological, that means the nosodes or sarcodes, then imponderabilia, allersodes, isodes. We will deal with each one of these, the botanical sources. Botanical sources comprised of the majority of the drug sources in homeopathy, and almost 60% of medicines are prepared from plant sources, and we are using the plant as a whole. Otherwise we can use in parts of the plant. Even we use the extract of the plant, the phytochemicals of the plant, etc. Now coming to the zoological sources, the animal medicines. So the medicines prepared from the animal sources. In homeopathy, we use the medicine prepared from the whole animal, whether in the live or in dried form, or the parts of the animals. So I'm not going to give some the examples. All of you know the examples. And sometimes we prepare medicines from the excretions or secretions of the animals, venoms of the animals, etc. Now coming to the minerals and chemicals. So in the mineral source, minerals comprise a large area of homeopathic medicines, where it contains the inorganic substances, organic substances, metallic, non-metallic, or mineral springs. So the medicine prepared from all these sources are used in as a homeopathic medicines. Now the unique sources of homeopathic medicines, these are called biologicals. Biologicals are it is classified into sarcodes and nosodes, and nosodes is the medicine prepared from the diseased parts or diseased secretions or diseased organs, whereas sarcodes are the medicines prepared from the healthy secretions, healthy organs, or healthy tissues of animals or human beings. So this is what is called as biologicals. Now coming to imponderabilia. Imponderabilia is one of the very unique source of homeopathic medicines, which are discovered by Hahnemann himself. Imponderabilia is a source of medicine which is prepared from the immaterial sources, the energy sources. For example, the sunlight, X-rays, moonlight, etc. Okay. Now the next source is allersodes. Allersodes are the medicines prepared from the allergy causing substances, the antigens, for example, pollen, dust mite, etc. Where a substance, if it is ingested or come to contact with you, if it produces some symptoms, then that is known as the antigens, isn't it? So the medicine prepared from the antigens are called as allergens. Now the last source is isotes. Isotes are the substances, the medicines prepared from certain substances which is used in the other systems of medicines, like conventional drugs, medicines used in modern medicines, or any excipients of the drug. Excipients means the raw material which is used to make the drugs or medicines. The what you call as the starch or magnesium stearate. These are all excipients in the preparing the tablet. So if the medicines are prepared from these excipients, then it is known as isotes. So this comprises of the sources of medicine: the botanical sources, zoological sources, minerals, biological sources, azotes and sarcodes, impondroplia, allergens, and isotes. Now coming to the next aspect of homeopathic pharmacy, that is a collection of drug substance. So there are some rules of collections and there are some relevance of the collection of drug substances. Why we are giving more importance for the collection of drug substances? So we should gather the raw material, the plants, not to destroy them. We have to generally gather the medicinal substances, the plants, never mix different species. For example, if you take a chamomilla, German chamomilla is there, Roman chamomilla is there, isn't it? So when you collect it, do not mix into different species. It should be separate. Gather only the healthy, clean plants. Do not collect a damaged plant. It should be healthy. Gather mostly indigenous plants, and that may not be possible in India because most of the polycrest medicine, most of the medicine used in homeopathic are exotic in nature, European plants. So it has to be procured. So as far as possible, we need to use indigenous plants, because indigenous plants contains the most biologically active principles. Discard the old unhealthy plants, so it should not be used. So these are the general guidelines for the collection of plant drugs. One more factor to be reminded while collecting the drug substances, especially the plant. Plant contains certain pharmacologically active substances like phytochemicals or phytoconstituents, and these phytoconstituents or phytochemicals are responsible for the pharmacological action of the drug. The phytochemicals such as alkaloids, glycosides, tannin, etc., saponins, then oils, volatile and fixed oils. So all these phytochemicals, these are responsible for the pharmacological action of the drug. So when you collect a plant, when you select a plant, you have to look for the concentration of the phytochemicals. So whether that particular plant should have the required amount of quantity of phytochemicals or phytoconstituents. So for that, certain factors has to be seen. The age of the plant, it should be adequate, neither young nor old. The climate and the soil. If a plant is collected from its natural habitat, that will be well and better, because it contains the required quantity of the phytoconstituent. Now the cultivations. So if the plant is cultivated by giving good manure and water, then that plant is a healthy plant. It contains the required amount of pharmacologically active phytoconstituents. It can be very well used. Okay, so these factors has to be considered. Now second is the collection of animal drugs. So I told you we are using the whole animals, the parts of the animals, secretions or excretions of the animals, even the venoms of the venomous animals like snakes. So the animal should be selected properly. There should be an expert in identification of animal drugs, like a veterinary surgeon. Get healthy animals. As far as possible, we need to examine the age and health of the animal, discard the unhealthy animals, test animal products of the parts, and collect genuine animal substances. I had experience in collecting several drug substances, especially from the animals like sarcodes. Once, when I was in industry, I was asked to prepare an nosot called pancreatinum, and I went to a place where the pigs are nurtured. With the help of a veterinary surgeon, we identify a healthy animal, cut it open, identify the pancreas, clean it, and a microbiologist has to certify that, because it should be free from the microbes or microorganisms. Once it is cleaned, it is free from all the microorganisms. Then we prepare the sarcode, pancreatinum. So it involves the expertise of several people: a zoologist or a veterinary surgeon, a microbiologist, a pharmacologist, etc. So this is what is called as a multidisciplinary approach in homeopathic pharmacy. Now coming to the mineral kingdom. So mineral kingdom, it should collect from the reliable sources. Suppose if you are purchasing a crude substance, a chemical from a vendor, a shop, it should be purchased either pharmaceutical grade or analytical grade. Pharmaceutical grade is good, and analytical grade chemicals are far better. It is excellent. Take utmost care for quantitative as well as qualitative assay of these chemicals. Do not mix the chemicals. Take precautionary measures. If it is a poisonous substances, there should be a word of caution how to handle these chemicals. Do not collect in the form of fine powders, because most of the chemicals are crystalline and white in nature. So we cannot identify by the mere look. So by purchasing a chemical in the powder form, if it is available only in powder, it's okay. But if it is available in other forms, in crystal forms or in other forms, we need to get it as the original conditions, not in the powder form. Okay, so this is what you have to bear in mind while collecting the mineral drugs. These are the general guidelines. Now coming to nosodes. So nosodes attracts a very special attention, because the nosodes are the product of the disease. The medicine prepared from the product of the disease. So it should be handled very carefully, because it is a diseased product. It contains a microbial organism. The microbes are available as pure organisms and obtained from suitable clinical material from subjects suffering from disease or isolated, cultured and identified. So normally we are classified the preparation of nosode into 4, that is N1, N2, N3 and N4. So what are called N1 nosodes? So in N1 nosode, the method of preparation says that it is prepared from the microorganism which produces the endotoxin. So there are some bacteria which produces the endotoxins. So such bacteria, such microorganisms are used to prepare the N1 nosodes, and it is the lysated, the killed bacteria is used, and this bacteria is used as a stock solution. The lysate, there are the specified methods are there. I'm not going to explain the method of preparation of nosode as such, but some general guidelines. Their properties are studied for a complete identification as per the individual monograph and their left side, and to ensure the preservation and stability of the characteristic. So I told you about the 4 methods: N1, N2, N3 and N4. Now N2 nosodes, these are the medicines prepared from the lysate of microorganism, which is prepared from the the microorganism produces the exotoxins. Now, N3 bacteria, N3 nosode. N3 nosodes are the medicine prepared from the purified toxin like tetanus toxoid. And N4 nosodes. N4 nosodes are the nosode prepared from the clinical material, what we call as the classical nosodes, which was prepared from herring or Hahnemann, etc. Like a sorinum, syphilinum, then medorinum, variolinum, etc. All these are classified under N4 because it is prepared from the clinical material. The disease product, the pus or the abscess or any exudate. So these substances are classified under N4 nosodes. Now, next one is sarcodes. Sarcodes, as you know, it is a medicine prepared from the healthy organisms, and the parts of the organisms, secretions. They should collect from the healthy animals, only from the healthy animal, because sarcodes means the healthy, fresh. Keep animals under observation. So before taking this, the parts of the secretions, we need to observe the animal for some days. Collect from the reliable sources. Endocrine secretions and few enzymes may be collected from cattle, sheep, from the slaughterhouses. So different secretions or different parts or even the whole organs are prepared for making sarcodes. Now coming to the next one, that is standardization. So we have identified the plant, we have collected it properly, we have preserved it. Now the next is standardizations. So what you are going to see in standardization? So there are three things to be considered. One is identity, quality, and purity. So what do you mean by identity? Identity is to identify the genuine species, the correct species of animal. So we have pharmacopoeia. We have different pharmacopoeias, official and unofficial pharmacopoeias. In India, we are using Homoeopathic Pharmacopoeia of India. So it is having 10 volumes. So these pharmacopoeia contain monograph, which is known as it is a what you call a detailed description of the drugs, which is known as monograph. And this monograph contain the guidelines for identification. So how to identify the correct species is described in the monograph. So you need to identify a species of the plant or animal, whatever it may be, according to the description of the pharmacopoeia. Next is the quality. So what do you mean by quality? Once you identify the genuine species, the correct species, next we have to check for the quality. So even though if you identify the correct species, it may not have the quality, required quality. So it should have an adequate quality. Should not be spoiled one, should be a healthy one, isn't it? So the quality should be assessed. Next is the purity. What do you mean by purity? We have identified a correct species and we ensure that it is having a very good quality, and if it contains any adulterants or impurities, what we will do? So that is why we need to assess for the purity. So the purity can be altered by adulteration. So we need to detect the nature of adulteration, the type of adulteration, and what substance is used. Sometimes it may not be adulteration, it can be a substitute. Many of the Indian brands, when I was in as a analyst in a quality control lab, we got some drug substance which are not the correct species at all. It is a substituted one, but it look identical. So an analyst should have a very good idea about the identification and the quality and the ability to detect the impurities or adulterants or substitutes. And another aspect of standardization is estimating the active constituents which is present in a crude drug. So what are I told you about the active constituents in a plant. These are pharmacologically active. These active principles are responsible for the pharmacological action. They are alkaloids, glycosides, tannin, saponins, oils, etc. And all these steps, identification, quality check, and purity check, it constitutes the evaluation of drug. Now we'll see what are the evaluation method we follow in homeopathic pharmacy. Organoleptic evaluation, microscopic evaluation, chemical evaluation, physical evaluation, and biological evaluation. Now we'll see one by one. Organoleptic evaluation is it is a method of evaluation by means of naked eye, the organ of senses, external morphology, shape and size of the raw material. So we can easily identify if the analyst is very expert. He can very well identify a species by just looking the drug substances, the shape, the size, even the external color, external marking, the fracture. Once you break it, how it fracture, how it breaks. So that determines the quality of the substance. The odor, sometimes, many times we need to smell it. Many of the herbs having a characteristic odor. Taste, sometimes we need to taste it and see. Many of the homeopathic raw materials used in as a homeopathic medicines, it is having characteristic aromatic taste, so we can easily identify it. The second one is microscopic examination. It is a method of evaluation by means of microscopically. That means we need to extract very small amount of extract. Then under the microscope we have to see. Then the powdered drug we can examine under the microscope and see the characteristics. The type of trichomes in plant drugs will give a vague idea about the what species it is, what genus it is. The histological pattern, the arrangement of phloem and xylem, the crystals, calcium oxalate crystals which is present, the oil components. So all these things can be seen under the microscope, which is known as microscopic evaluation. The microchemistry, even we can extract the drug substance and see under the microscope and see the constituents. Crystallography, these extracts can be converted into crystal, and the pattern of crystal which forms will tell you which species it is. So these type of technologies, that is why I told that homeopathic pharmacy is a multidisciplinary approach. It's a method. So all these technologies are used for the standardization. Now coming to the next one is chemical evaluations. So in chemical evaluation, what we are going is we have to isolate it. Isolate the drug, identify it, purify it, then characteristic determination of drug having active principles. So all these active principles have a specific test. Test for alkaloid, test for glycosides, test for tannins, and there are some quantitative assays. So we can identify the presence of the phytochemical and even the quantity of phytochemicals by means of different instruments like UV spectroscopy or HPLTC. All these methods we are using. So now coming to the chemical method, the first one is color reaction test. So what color it produces? If you add some reagents, what color it produces? So that will give a characteristic result. Acid value, iodine value, saponification value, ester value, ash value, sulfated ash value, then acid insoluble ash, water insoluble ash. All these are specific parameters for identifying the drug substance. So this can be used for identification. So the next one is physical evaluation by means of various instruments, sophisticated instruments like chromatography. Chromatography can be TLC, simple chromatographic method, TLC, or HPTLC or HPLC. These are advanced chromatographic methods. Fluorescent analysis is another method. Solubility, specific gravity, melting point, congealing point, refractive index, optical rotation, water content or loss on time. So all these things are called as physical evaluation, which are specific to each and every drug substance. So it can be used. Now the biological evaluation, this biological evaluation is another method of evaluating the drug, especially the activity of the drug. So the biological evaluation can be done in the intact organs or tissues or in animals. So that is called as animal studies. So these assays on living animals and on their intact or cut organs to indicate the strength of the drug or their preparations. So as living organisms are used, these assays are known as biological assays. So animal experimentations are there. Now coming to the vehicle, we have classified the vehicles. Vehicles are the substance used for transport or conveying the dynamic energy from the medicine to the interior of the organisms. So vehicles can be used for extraction purpose. Vehicle can be used for preservation purpose. Vehicle can be used for making potencies. Vehicle can be used for making storage, preservation, etc. and for dispensing. So vehicles are classified into solid vehicle, liquid vehicle and semi-solid vehicles. Here the solid vehicles are sugar of milk, cane sugar, grape sugar, globules, cones, tablets and pellets. So these solid vehicles are used to make triturations or potencies or mother substances, and it also can be used for preservation of medicines. These solid vehicles can be used for dispensing the medicines. Similarly, the liquid vehicles are classified as purified water, alcohol, and different types of alcohols are there. Glycerin, simple syrup, oils, fixed oil and volatile oils are there. Then coming to semi-solid vehicles. Semi-solid vehicles are mainly used for making external applications, especially the Vaseline, beeswax, spermaceti, lanolin, then soap, vegetable hard and hard soap, soft soap, curd soap, prepared lard, isinglass and stuff. So these semi-solid vehicles are used for making external applications. I'm not going in detail about these vehicles. Coming to the extraction method. So generally homeopathic medicines are extracted with mainly two methods, that is the old method or Hahnemannian method or classical method and the new method. The old method or Hahnemannian method was originally devised by Hahnemann for preparing mother tinctures, and in old method Hahnemann understand that each drug substance, each plant having its own moisture content. So depending on the water which is present in the plant drug substance, it varies, the method varies. So he generally classified the methods into three: mother tincture, mother solution and mother substance. And here mother tincture, there are 9 classes for old method. In mother tincture, class 1 to class 4. In class 1, Hahnemann classified the drug substances which is having more juicy. Most juicy drug substances are there. Then class 2, moderate juicy. Class 2, least juicy. And class 4, it is dried drug substances. Now class 1, what do you mean by class 1? As I told you, it contains more water content, most juicy plant. And the drug menstruum ratio, the vehicle which is used to make extractions or mother tinctures are called as menstruum. So generally we use alcohol water mixture. So the drug menstruum ratio in class 1 is 1:1, just equal part. The drug strength is naturally 1/2. And the example, the fundamental rule, Hahnemann has explained about this class 1 in belladonna, under belladonna in Metra Medica Pura. Class 2 in similar way, the drug menstruum ratio is 3:2. That is 3 parts of drug substance, 2 parts of menstruum. And the drug strength is 1/2. Drug strength means the actual amount of actual quantity of the drug which is present in a particular product. That is called as drug strength. So the drug strength in class 2 is 1/2. The fundamental rule, Hahnemann explained about class 2 under Tuja in Metromedica pura. Now class 3. Class 3 contains least juicy plant and drug menstruum ratio is 1:2, one part of drug substance and 2 parts of menstruum. And the fundamental rule here is Villa. The drug strength is 1/6. Next is class 4. Class 4 is a dried drug substances. So it the ratio is 1:5, one part of medicine and 5 parts of menstruum. And the drug strength is 1/10. And the fundamental rule here is Vigilia and Staphylococcus. So next, coming to mother solution. So mother tincture is the plant extract. Is extracted from the plant. Whereas mother solution is mainly prepared from the chemical or mineral origin. So it is just a solution in water or alcohol. It is not extracted. It is just a solution. That is why it is called as mother solution. Another one is mother substance. Mother substance is the powdered form, the triturations. The substance which are not dissolving either in alcohol, in water, it has to be triturated. It has to be grinded with the help of mortar and pestle by using sugar of milk. So those substances are called as mother substances. Now coming to mother solution, we are having two methods, that is class 5 and class 6. Again, it is classified 5/6A and 5/6B, class 5B. So in class 5A, the substance which are soluble in less quantity of water, for example, sodium chloride, natrium muriaticum, that is an example for class 5. Another one is class 5B. In class 5B, it requires more amount of water to dissolve. So class 5 is the substance soluble in water. Class 5A, it requires small amount of water to dissolve it. Class 5B, it requires large amount of water, more quantity of water to dissolve it. Example, acid phosphoricum. So here the drug strength in class 5A is 1/10 and class 5B is 1/100. Coming to class 6. Class 6 is mainly the drug substance which are soluble in alcohol. And here also it is similar way. It is classified into class 6A and class 6B. And 6A, it requires small amount of alcohol, and in 6B it requires a large amount of alcohol. So here the drug strength is 1/10 in class 6A, and class 6B it is class drug strength is 1/100. Now coming to the mother substance, class 8. Class 8 is dry insoluble substances. That is, it is a solid in nature. It is dried drug substances, but it will not dissolve in alcohol and water. So it has to be triturated with sugar or milk. So this can be prepared with the decimal scale or centesimal scale. That is 1/1:9 ratio or 1:99 ratio. So classical example is arsenic amalgam. Now class 7, class 8. Class 8 is a liquid insoluble drug substance. The nature of drug substance is liquid in nature, but it is insoluble in water or alcohol or any other liquid solvents. So it has to be triturated with the sugar of milk, and it is classified under class 8. So most of the nosodes are prepared with the class 8 in old method. Now coming to class 9. Class 9 is a fresh drug substance from plant or animal origin, and here the ratio is 2:9 and 2:99. Being it is a fresh drug substance, during the process of trituration, the one part of the drug substance may be evaporated, contain the moisture. So the ultimate drug strength, the actual quantity of drug will be 1/10 or 1/100. And the classical example, the fundamental rule here is Agaricus muscaris. So this is a general outline about the extraction method in the old method or Hahnemannian method. Now coming to next one, that is a modern method. So why modern method? Hahnemann considered the moisture is a part of the drug substance, and in Hahnemann's time, majority of the medicines used by Hahnemann was indigenous to him. It is a natural habitat is Germany, so or in Europe. But when homeopathy grows globally across the world, then most of the countries has to procure the drug substance from different countries. So it was difficult. So that was observed by homeopathic pharmacopoeia of United States, and previously the thought process was coming in British homeopathic pharmacopoeia, and they made some slight changes and they decided to use the solid substances, that is the powdered form, as a starting materials. So that is how the modern methods have come, and it has got two method, maceration and percolation. So maceration is a very simple process, but it requires more time to extract, 2 weeks to 4 weeks to extract. And the nature of drug substance used in maceration is a gummy, mucilaginous substances, which is very difficult to extract it. So the menstruum, the solvent and the drug substance has to be in contact for more time. More time is required to extract the active principle from the drug substance. So that is why maceration is a process of a long process. Whereas percolation, the next method is percolation, where we are using the powdered drug substances, which are very easy to extract, and it requires less amount of time. 24 hours to 48 hours is required for percolations. And here what happens is the menstruum is percolated through the drug substance, so the extraction is very easy. Okay, so here the gravity plays very important role, then surface tension, osmotic pressure, etc. These forces which retain the menstruum to the drug substance and facilitate the extraction processes. And here the difference is consideration of moisture content. So once you consider the moisture content prior to the preparation of drug, and accordingly we calculate the monograph formula. So what do you mean by monograph formula? The formula for making drugs, preparation of drug, which is given in the monograph in the pharmacopoeia. So it contains water, alcohol, or even glycerin. That depends on the drug substance. Consideration of dry crude as starting material, as I told you, in new method, we consider the dry crude substance, the powdered drug substance, as a starting materials. And one more thing is uniform drug strength. So when you use 100 grams of powdered drug substance as a starting materials, naturally the drug strength is 1/10. And there are few exceptions for that. Like phosphorus 1/667 or sulfur 1/5000, Moscus 1/2, 1/20, caustic 1/2, etc. So these are some exceptions other than 1/10 or 1/100. So that is about the modern method. Now coming to potentization. So far I am discussing about the very common thing which we followed in the homeopathic pharmacy, like selection, identification, preservation, preparing the extract, etc. Now coming to an area where we can see the uniqueness of homeopathy. So this is the area we need to study more, we need to research more, and we need to explore more. So that is what is required for the concept of potentization. So before going to that, I'll explain about some conceptual things in the potentization. First one is the scales of potentization. So here we have 3 different scales for preparation of the medicines, that is decimal scale, centesimal scale, and 50 millesimal scale. So decimal scale is ultimately, it is 1/9 ratio, one part of drug substance and 9 part of vehicle. It was originally devised by Herring, Constantine Herring, to prepare the snake venoms and other lower potencies. And normally decimal potencies are designated as X or D, and still it is used to prepare biochemical preparations, lower dilutions, etc. Now the concept of decimal potency is every succeeding potency contains 1/10 part of the preceding one. That is a concept of decimal potency. Now Next one is centesimal potency. Centesimal potency was originally described by Hahnemann. It was given in 5th edition of Organon of version 270. And here in centesimal potency, the drug menstruum ratio is 1:99. That means every succeeding potency contains one part of preceding potency. Okay, and it is usually denoted by C, CH, or simply numericals. So this is about the two scales. Now the next one is 50 millisimal potency is the last elevation of Dr. Hahnemann, which was published in the 6th edition or the last edition of Organon in a version 270. And Hahnemann discovered 50 millisimal potency after many trial and error, and he concluded that this is the most appropriate potency, and he termed it as a new, altered but perfect method of potentizations. But 50 millisimal name was given by P. Smith of Geneva. Okay, so this is the preliminaries of 50 millisimal. But the clinical application of 50 millisimal is it is highly effective potency for dealing with any type of disease, whether it is an acute or chronic diseases. I personally use 50 millisimal potencies in many occasions on most of the cases. And one of the feature of this, one of the speciality of this potency is it having minimum aggravation and maximum efficacy. It can be used in all type of diseases like mental diseases, any type of chronic diseases, one-sided diseases, etc. So it is having a highly latent power of medicines. So method of potentization. So now we discuss about scales. Next is the method of potentization. Now once the extract or mother tincture is made, next we need to prepare the potencies, homeopathic potencies. In modern term, we use ultra high dilutions, ultra high dilutions of homeopathic medicines. So now it is the time of ultra high dilution. I will come to that later. What is the relevance of ultra high dilutions? So two methods, succussion and trituration. Succussion is a method which is used when the drug substances are soluble in nature. When the drug substances are soluble, we use succussion method. We get you first prepare the mother tincture, then we dilute with the vehicles successively and serially while giving a downward stroke. That is succussions. So 10 succussions are given. So one part of the drug substance is taken and 9 part of alcohol or whatever vehicle we use. Then give 10 succussions, we'll get one potency. This process is continued to get the desired amount of potency. So normally in centesimal scales we use a 30 potency, 200 potency, 1M, 10M, 50M and CM. So these are the chronological order of the centesimal potencies. Now if the drug substance is insoluble, so we need to triturate, we need to rub with the help of sugar of milk in mortar and pestle, and the powdered drug is prepared up to 6x potency in decimal. Then later it should be converted into 8x potency by means of fluxion potency. That is one part of 6x, 1 grain of 6x is taken and it should be mixed with 50 parts of water. Then again it should be mixed with 50 parts of alcohol. Then give a 10 succussion, will get a 8x potency, skipping the 7x potency. Similarly, in centesimal scale also we can prepare. We prepare up to 3C potency of trituration, insoluble drug substance. Then this 3C should be converted into 4C by the method of fluxion potency. So this is how it is converted into a solid trituration is converted into a liquid potency. So that is about succussion and trituration. Now my interest in potentization is what is the drug substance to cure the disease. So what is the material content? What causes the cure process in homeopathic potencies? So recent revelations says that potentization is nothing but it is a classical top-down nanofabrication. So nanofabrications, many of you might have heard about nanofabrications. It is about fabricating the nanoparticles or quantum dots in a nanoscience. So there are different methods for nanofabrication. That is according to nanoscience. A top-down method is there. Then bottom-up method is there. And even in top-down method, chemical-induced, physical-induced methods are there. Proton banking method is there. So here, according to Nanodynamics, the book written by E.S. Rajendran, he claims that the method which was discovered by Hahnemann, the classical potentization method, by mixing one part of duct substance to the 9 parts or 99 parts of vehicles and give the downward stroke, succussions, strong succussions, equal succussions, it mimics exactly with the classical top-down nanofabrications. So what substances, the substances, which is present, the biologically active substances which is present in the higher and higher ultra dilutions of homeopathic medicines, these are nothing but the quantum, the nanoparticles or quantum dots, which are biologically active, pharmacologically active. Okay, so the first of this study has appeared in 2010, which was done by Bellare and Suresh etc. Et al. And extreme homeopathic dilutions retain starting materials and nanoparticulate perspective. This is the first study, as far as I know, regarding the concept of nanoscience in homeopathy. And later, the same people, they've done another study, just published in 2016, and very recent study was published, or the compilation of all these studies are published in 2019 by Dr. S. Rajendran, Homeopathy seen as personalized nanomedicines. So this concept is a very new concept in homeopathy, and as far as possible, it can effectively rationalize the method of potentization. Now, we detect the nanoparticles or quantum dots in homeopathic potencies and how it interacts with the body. What is the biological interaction with the nanoparticles in the body? So there are another studies which lead by Dr. Kudabaksh. He found that some epigenetic modifications are happening with the potentized homeopathic medicines. So what do you mean by epigenetic modifications? Epigenetic modifications are the modifications at the level of gene, where it down regulates or upregulates the gene functions. So such epigenetic modifications are ultimately expressed in the form of cure in homeopathy. So now there are a lot of studies. Which takes place how the disease caused, especially in cancer cases. It is nothing but some errors in the protein synthesis or protein uptake, which is ultimately the error in a DNA or in the gene. So it can be only corrected with the epigenetic modifications or the upregulation of gene, the affected gene. So these can be achieved only by the very small amount of drug substance at the level of nanos. So that is what is called a homeopathic potentized homeopathic medicine is otherwise known as the biologically active nanoparticle or quantum dots. So this is the only substance which can subtly touch the genetic factors or genetic codes which causes the epigenetic modifications. So a lot of studies have come up after that, and I selected only few references for this. That is the two references. Contrago 30 induces epigenetic modifications of lung cancer, specific tumor suppressor gene via demethylation. So DNA methylation is the key point here. Another study also conducted in hydrastis and chondrogo potencies, ultra-dilutions, which induces epigenetic modifications and alter the gene expression. Even in drug proving also, when you take the medicine, when the prover is taking medicine, what causes is a transient genetic gene modifications, which can be reversible. Okay, so that is what is given in the reference books and the journals. Now coming to the drug development in homeopathy. So all of you know that in homeopathy, drug development means drug proving or homeopathic pathogenetic trial. So before going in detail about the drug discovery or drug development process, let us see how a drug is developed in modern medicine. There are mainly 4 phases for that. First one is the initial synthesis and characterization, where we identify a group of drug molecules, and it has to undergo a lot of assays, like a lot of screening assays, like high throughput screening, ultra high throughput screening, etc. And we find out some hit compounds, what are the leading compounds, and we characterize it. So the substance has to be physically and chemically characterized, the structure should be elucidated, and later once the substance is identified, next we go to the preclinical studies, which comprised of in vitro studies, that is the studies which is conducted in the laboratory in tissues or in some medias, and later the dose is defined. Then comes to the in vivo study, that is the animal study. Okay, once the drug is studied in animals, lower animals, we fix the minimum recommended screening dose, maximum recommended screening dose. So this is the transformation from the animal dose to the human dose. So maximum recommended screening dose to the safety screening dose, that is SSD. So once we achieve the safety screening dose, then we can go for the clinical trials. And here in clinical trial, there are 3 methods: phase 1, phase 2, and phase 3. So in phase 1, the medicine is given, the drug is given in healthy volunteers, and checking for any symptoms appears after giving the medicine. So mostly adverse reactions, side effects of the drug, they are expected. But there are some similarities between the homeopathic pathogenetic trial or drug proving and the phase 1 drug trial. So here the similarity is both in phase 1 clinical trial and homeopathic pathogenetic trial, we use healthy individuals, healthy volunteers, and the difference is in phase 1 clinical trial we expect less amount of symptoms because we are expecting the adverse drug reaction, side effects, whereas in homeopathic pathogenetic trial or drug proving we expect more symptoms, more refined symptoms, more subjective symptoms. So these are the differences and similarities between phase 1 and phase 1 clinical trial and homeopathic pathogenetic trial. Now coming to phase 2. Phase 2 is efficacy study. We are selecting some patients in a small group, giving the medicines, and we check for the efficacy as well as the side effects. Now the third phase is for a large number of patients. In a real situation, we test it, and mainly we expect the effectiveness of the drug. Okay, so now after that the new drug application has to be reviewed by the regulatory bodies. Then the drug is released in the market, and the next phase is called as post-marketing surveillance, or called as phase 4 study. So what is the purpose of or objective of post-marketing surveillance? Post-marketing surveillance, the adverse drug reaction, the reporting of adverse drug reaction is expected. So how many symptoms are obtained as a side effect? So that has to be analyzed. So this is what is happening in post-marketing surveillance. So this is a fourth step for new drug development in modern medicine. Now coming to the drug discovery in homeopathy. Now we call it as homeopathic pathogenetic trial. Homeopathic drug proving is known as homeopathic pathogenetic trial. And the study design here is prospective, double-blind, parallel, randomized placebo-controlled study. So here prospective double-blind, that means it is a prospective study. Double-blind, even the volunteer, the subjects, and the experimenter is not knowing about the nature of drug or placebo. Parallel, the both drug, the Viram group and placebo group are running together. The test is done in both arms. Randomized, here the medicine and placebo is given randomized. Placebo controlled, placebo also is involved in this study to differentiate the effect of placebo and the drug. Now the endpoint is classified as the disappearance of symptoms during proving and subsequent symptom-free period for 30 days. So this is the endpoint where we stop the symptoms. Now coming to the methodology of homeopathic pathogenetic trial, the first step is the protocol. As per the protocol, the volunteers, the healthy volunteers are screened. There are some criteria for that. Some qualities of drug provers, there should be healthy, apparently healthy, trustworthy, delicate, etc. These are the some and there are some inclusion criteria and exclusion criteria. Then after that, the pre-medical test is pre-trial medical test, that is PM is done. So all the laboratory parameters, all the health checkups, everything is done, and the patient is certified as a apparently healthy patient. Now the next one is enrollment as volunteer. Once all these tests are done, later then the volunteer is enrolled as a prover. Next step is the run-in period. So here the running period means the patient has to be observed, self-observation is done for 2 weeks. And at the time of running period, they have to write the symptoms. And after the running period, we need to give there is an interventional phase. So we need to give the medicine or placebo, whatever it may be. And the dose in this is the CCR, Center Council of Research in Homeopathy protocol. And I am the co-site investigator in one of the site. It is a multi-centric trial. So I am the co-site supervisor in one of the center of CCRH drug proving. So here what we are going, the dosage is 12 doses we are giving, 4 doses for 3 days. And the duration of taking medicine is either 3 days or when the symptom starts. When the symptoms start appearing, we have to stop the medicines. So now the symptoms appears, and the volunteers are asked to record the symptoms in a specified manner. Then they need to observe until the symptom persists. Then we need to go for the assessment of any adverse reactions. So sometimes we never reported any adverse reactions normally because we are using the ultra high dilution homeopathic medicines. Now suppose the symptoms disappears, then again the volunteer is kept for a washout period without giving any medicines for 4 weeks. And during the washout period also, they need to observe self. If there any what you call as transfer effect of drug is there. Now suppose if there is no signs and symptoms after the intervention, maybe because of placebo, or even the placebo group also exhibit some of the symptoms which is identical to the medicines, that is psychological. And suppose if the volunteer is not experiencing any symptoms, then again the volunteer has to go for a washout period. Then if no symptoms in the washout period, then the volunteer can be taken for a post-trial medical examination. So the medical examination, including all the checkups and laboratory investigation, should be done before the starting of trial, the drug proving trial, and after the pathogenetic trial. So this is the outline of the process, the procedure, or the protocol of the drug proving project of CCRH.
Sir?
Now, the recording of symptoms. Yes.
So are we near the end of the session?
Yeah, a few more slides are there.
Okay. Sir.
Okay. I will make it fast.
Okay.
So now the characterizing feature of proving symptoms, new symptoms with marked severity, duration and frequency, ongoing or recurring symptoms present during the proving, ongoing or recurring symptoms that have been unexpectedly marked or worsened, symptoms that require from the past but not have occurred 12 months preceding the proving, that is, the symptoms is not experienced before. Symptom display alternation with other symptoms in a single volunteer in such a way that alternation is strongly individualizing, so individualizing symptoms. So here the care should be given to the time of onset, duration, then the location, sensation, then modality, extension, etc., and concomitant. So this is the order by which the symptom has to be recorded by a volunteer. Now, another aspect of homeopathic pharmacy is pharmacovigilance. So this is a evolving branch of or aspect in homeopathy. That is, it is defined as the science and activities relating to the detection, assessment, understanding, and the prevention of adverse drug effect of drug-related problems. So normally, homeopathy is considered as a system of medicines with no side effect. It is not like that. There are many side effects are there, which is not, we are not percepting that symptoms. Okay. Now there are some methods, some protocols to how to understand the side effects or adverse drug reactions of homeopathic medicine. So this can be due to phytochemical interaction, especially if we are using a mother tincture for a long time in a large quantity. Phytochemical interactions are there. Or if you are using ultra high dilutions or mother tinctures, medicinal aggravation may be one reason for adverse drug reactions. So how to assess the adverse drug reactions? So there are some assessment terms which we use to consider whether it is an adverse drug reaction or not. So first one is certain. Certain means, suppose if you take a medicine and we appear an adverse drug reaction or side effects, so we assume that it may be certain. It may be because of the drug taken. So plausible time relationship will be there. Plausible time relation with the drug and symptoms. So once you withdraw the drug, the symptoms may disappear. That is called as de-challenge. Then when you restart the medicine, again the symptoms start appearing. That is called as re-challenge. So here, if a adverse drug reaction is termed as certain, it should fulfill the de-challenge and re-challenge criteria. Now the probable, suppose the ADR or adverse drug reaction is probable, then only the de-challenge, there will be a time plausible relation will be, there is an exact definite relation between the drug and the symptoms, but only de-challenge. Once you withdraw the symptoms or withdraw the drug, then the symptoms will be removed, and re-challenge is not applicable in probable. Once you re-administer the drug, may not appear, the symptoms may not appear. Possible, there may not be a de-challenge. Once you there may be a chance because of the drug, the symptoms may be by chance. It can be due to a concurrent disease or maybe because of other drugs. So there is a chance, that is called as possible. Unlikely means there is very less chance of being due to the drug. The adverse drug reactions are very unlikely due to the particular drug. Okay, and there is no re-challenge and there is no de-challenge. It can be maybe. Condition or unclassified means there may be other reasons or the assessment cannot be possible. So conditional or unassessable can be used simultaneously. So these are some terms which is used to describe the pharmacovigilance or the adverse drug reactions in homeopathy, in AYUSH. Now coming to the last few slides, these are some drug laws. I'm not going to detail about this. So this is not so relevant. I'll go to next. So this is very important. So fundamental research in homeopathy comparison. So there was a systematic review which has happened in 2010 and also in 2015. So the result, I just quoted only the result part of it. In 2010, the methodological quantity quality of the generally adequate. That is, this is I am comparing two studies, two systematic reviews in the field of animal studies with homeopathic drugs in two different time period. Okay, so in up to 2010, the methodological quantity was generally adequate, but some aspects should be improved. That was a conclusion of the authors. Convergence between the result and mathematica is seen in some studies pointing towards the possibility of systematic study of similar principle. So there is some possibility. Effect of higher dilution seem to be stimulate restoration of stable state, what we call as the homeogenesis, as seen in several experimental models, both descriptive as well as a mathematical point of view. So in 2010 study, the method was just adequate. And the same people, same researchers, they did another systematic review in 2015. So the conclusion was very encouraging. The conclusion was the quality of the employed techniques improved previously compared with the studies that were reviewed previously, with the inclusion of more ethically refined protocol. That is very, very important. Ethically refined protocols. Within this first 5 years, the protocol, there are a lot of changes in the protocol, including in vitro primary cell cultures and in vivo studies, often with 3 or more replicates. So reproduction of the result is very, very important in research. If you cannot replicate any research study, that research is not valid. So we can replicate the 3 or more times. And analysis of epigenetic mechanism that were previously unknown in 2000. So see, within these 5 years, how much advancement was there in the research, especially the fundamental research in homeopathy. So that is very, very encouraging, very, very encouraging. And that is why I am having an appeal to the scholars, since Yenepoya University has started the PhD programs. And I presume that the students who studied in the college should have a research bent of mind. So an appeal to them. Homeopathy is a process of evolution from a holistic alternative system of medicine to a personalized nanomedicine. That is very, very important. The dimension of homeopathy is going to change within few more years. So a stringent, precise and robust research protocol should be required. And all the students and scholars of homeopathy should contribute to the development of the system. And you should identify your own space. And this is my appeal to the scholars and students of homeopathy, especially the teachers, the faculty members, and the policymakers. Thank you. This is the end of my presentation. And I thank the management, dean, the faculty members and students of homeopathic, Yenepoya Homeopathic Medical College, and thank you once again.
Thank you, sir. It was a very elaborative and very knowledgeable session. Now I have a question from one of the participants.
Yes.
Can you please explain the layers of percolation?
Layers of percolations. The percolator is an instrument, it's a conical instrument. Is it audible?
Yes, sir, it's audible.
Yeah, it's a conical instrument which is having a head and a body and the lower part. And here, the lower part is covered with a porous substances called toe. And above that, there should be some layers. The purpose of these layers of sand or glass is to retain the vehicle with the drug substance. Because in percolation, what we are doing is 24 hours. There is a misconception in the percolation that it is the process of percolation require only 24 hours. It is not like that. 24 hours we have to keep the vehicle and drug substance together. We have to retain it. Then after that, the extraction process. So to retaining the drug substance and the vehicle, we need to have some obstruction, but at the same time, this obstruction should allow the vehicle to percolate through the drug substance. Understood. So the layers are, it is depending on the sand or a glass, that is one by half inch of sand is placed, that is a coarse sand, then medium coarse sand, then fine sand. So there are 3 layers in that. So these layers keep the menstruum as well as the drug substance together for 24 hours, so that it can percolate, the what you call as the solvent can percolate to the interior of the drug substance, the powdered drug substance, and facilitate the extraction. So that is the arrangement of the obstruction in percolation.
Okay, sir. There is one more question from one more participant. Please explain how monograph formula was written in HPI book.
Monograph formula is mainly for preparing the mother tinges. So how we are preparing the monograph formula? Suppose in new method we are using the powdered drug substance, dried powdered drug substance as a starting material, isn't it? So now in fresh form, how much moisture is present in the fresh form? That is to be compensated in the monograph formula. So monograph formula contains alcohol and water. So the amount of water is calculated on the basis of the moisture estimation. So while in fresh, how much moisture is present, and that quantity of water should be present in the monograph formula. So this is how it is calculated. So for that, what we have to do is we need to procure the fresh drug substance, find out the moisture, and convert it into percentage, and that percentage of water should be added to the monograph formula. So this is how it is calculated. The objective is the actual amount of water which is present in the fresh form should be there in the monograph formula. Follow?
Yes, sir. There is one more question. Why green glass is used in percolation or modified laceration process? Why not other glass?
What is that?
Why green glass is used in percolation or modified laceration process? Why not other glasses? Please explain.
That I don't know exactly why green glass, but glass pieces are used because glass pieces has to be triturated with the drug substance. If the drug substances are very fibrous in nature, where it contains more of mucilage, it has to be triturated with the glass pieces. So what happens is the minute glass powders will enter into the cell structures of the powdered drug, and it facilitates the percolation process, extraction process. But why we are using green glass, I am not having any idea.
Okay, sir. I think that is the last question. Now, I request Dr. Shilpi Rastogi, Professor and Head of the Department of Anatomy, to deliver the vote of thanks.
Thank you, Dr. Sushrut. On behalf of Yenepoya Homeopathic Medical College and Hospital, I, Dr. Shilpi Rastogi, Coordinator for National Webinar Series, take this opportunity to convey a hearty thanks to Dr. Ram Jyotis sir for such an informative session on an important subject of homeopathic pharmacy. Thank you, sir.
Thank you.
I would also like to place on record thanks to all the participants all across the nation for joining us for this learning venture. Finally, I would like to extend my warm thanks to all the representatives involved in organizing the event. Thank you, one and all. Over to you, Sushrut sir.
Thank you, madam. So there is one more question from one of the participants.
Yeah.
Drug development in homeopathy includes only drug proving or clinical verification too?
Yeah, in homeopathic pathogenetic trial, um, the drug proving is the main part. But one thing is that we need to characterize we need to define the proving substance. It is called as investigational proving substance, IPS. So characterizing of IPS is the preliminary part of drug proving, like modern medicine. So characterizing of IPS is done in many ways. It can be done by characterization. Chemical characterization should be chemically defined or physically, and should be physically defined. Then the pharmacognostical study has to be done. It has to study in vitro.